Hello everyone, my name is Khushbu Kushwaha. I'm working as an assistant professor in BBD University, Lucknow. Today I'm going to discuss about plant tissue culture. So let's start. Introduction to plant tissue culture. Plant tissue culture is a powerful biotechnological technique that involves the controlled growth and multiplication of plant cells, tissues, and organs in sterile environment. This revolutionary method has numerous applications in agriculture, horticulture, and plant conservation. Plant tissue culture is also a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterilized condition on a nutrient culture media of known composition. Plant tissue culture is widely used to produce clones of a plant in a method known as micropropagation. Now, principle of plant tissue culture, it include totipotency, aseptic condition, and nutrient media. Totipotency, the ability of a single plant cell to regenerate into a complete organism. Aseptic condition, maintaining a sterilized environment to prevent contamination of culture. Nutrient media, providing essential nutrient, growth hormones, and energy source for plant growth, as it is shown in the picture. Now, common techniques and equipments. These are the techniques which are used in plant tissue culture, that are explant preparation. Explant preparations are any part of a plant taken out and grow in a test tube under sterilized condition in special nutrient media is called explant. Media propagation, inoculation, incubation, and subculturing. These are the techniques through which we are grow plant tissue culture. Equipment which are required in this culture, that are lamina, flow cabinet, autoclaves, incubators, bioreactors, and analytical instruments. Precisions are carefully handling and aseptic techniques are crucial for successful plant tissue culture. Callus inductions and plant regenerations. First, callus induction. Callus induction is a redifferentiation of plant cells to form a mass of unorganized cells. Callus culture is also a production and maintenance of callus on a solidified nutrient medium under aseptic condition. Callus is nothing but a undifferentiated mass of a cell. It is given in a figure. Organogenesis, it is an induction of shoot and root formation from the callus. Embryogenesis is the formation of somatic embryos directly from plant cells or tissue. Next, micropropagation and clonal propagation. Micropropagation is a rapid multiplication of plants from small explant such as meristem or axillary bud Micropropagation is an artificial process. It is an advanced vegetative propagation technology for producing a large number of transplants in a limited time and space. Clonal propagation is a multiplication of genetical identification copies of a culture by asexual reproduction is called clonal propagation. It is also a production of genetically identical plant through vegetative propagation, and its advantage is high multiplication rate, disease-free plant can be produced, and prevention of desirable traits. Now application of plant tissue culture. It is useful in agricultural, horticultural and conservation. Agricultural use in crop improvement. Disease-free plant is produced through this technique, and germplasm conservation. Horticulture is rapid propagation of ornamental plant, virus eliminations and hybrid plant produce. Conservation, preservation of rare, endangered and threatened plant species. Type of plant tissue culture. Type of plant tissue culture, it includes organogenesis, embryogenesis, meristem culture, cell culture. Organogenesis is induction of organs like shoots and roots from plant tissue culture. Embryogenesis is formation of somatic embryos directly from plant cell or tissue. Meristem culture is nothing but a rapid propagation of disease-free plant from shoot apical meristem. Cell culture is growth and manipulation of isolated plant cells, tissue or organs. Now, nutritional requirement for growth of plant tissue culture. Nutritional requirement of plant tissue culture, it composition of nutrition normally depend upon particular species of plant or the part which part we are using for plant growth. First requirement is macronutrient. Macronutrient is required in large quantity greater than 30 ppm. It is essential. It's essential element like nitrogen, phosphorus and potassium. Nitrogens help in growth and development of plant. Magnesium is used for part of chlorophyll, and calcium used for cell wall formations. Micronutrient is nothing but it required in small quantity and its concentration is less than 30 ppm. The trace element like iron, boron and copper, these used for enzyme synthetic. Growth regulator, it include auxin, cytokinin and other hormone to control plant development. Auxin is help in cell division, root formation and growth of plant from the tip. Cytokinin help in cell division, shoot formation. Auxin help in root formation and cytokinin help in shoot formation. And the other growth hormones are gibberellin, abscisic acid and ethylene. Ethylene is nothing but induction, used in induction. Thank you.